Rent Mason Bees is committed to helping mason bees thrive in their environment. Through our partnerships with universities and research teams, we are able to support pollinators who are critical to our ecosystem and food supply. Pollinators are responsible for 1 in every 3 bites of food we take, so it's really important for us to understand our little solitary bees and make sure that we're taking really great care of them in our environment and studying those predators that are harming them, which is what we're going to share with you in this video. Solitary mason bees are solitary. It means they live alone, work alone. Each female lays all her own eggs. She finds all her own food and has to find her own nests. They do not have mandibles that are strong enough to chew wood, so they have to find natural holes in their environment or use your nesting blocks. They are some of the sweetest bees out there. They don't sting, they're non-aggressive, and it's our job to help and protect and make sure that we are releasing healthy bees into the environment and teaching all of you how to release healthy bees into your environment too. So with this research, we partnered with UC Riverside to study pollen mites and chalkbrood, and it's pretty fascinating what they've found, which I'm going to share with you in this video. We partnered with Jessica and Jacqueline that work in the Department of Entomology in the McFrederick Lab. In the fall, we harvest and clean millions of mason bee cocoons. When we do this process, we also find millions of predators, Houdini fly larvae, chalkbrood, pollen mites. There are a lot of really bad things that can harm our solitary bees. You can check out our YouTube channel and watch our harvest video. It's pretty fascinating to see all the steps that we take to make sure that all of the predators are removed from our solitary bees. This year alone, we cleaned over 3 million mason bee cocoons. And it's really important, and I always say this in our videos, to harvest and clean your cocoons because there are so many predators inside those nesting blocks. And on the cocoons of the mason bees. So Houdini fly larvae, those little flies will wait outside the mason bee block. She'll wait for the mason bee to leave, and then she'll fly in and deposit all of her little babies. They're called kleptoparasites, so they're going to eat the food supply that the mason bee left for her baby. And the mason bee doesn't even realize that those predators are in that cell. She'll cap that cell off with mud, and then those little babies, those Houdini fly larvae, will stay in there all winter long, and they'll emerge with mason bees the following spring. Pollen mites are the same thing, except for those little suckers. You wait till you see them under the microscope. I'll share that with you in a little bit. But pollen mites multiply very rapidly, very quickly, and so they will also go in and get deposited in the cell, and they'll eat all the pollen, and they'll eat the larvae and eat the pollen that was left. And then when the mason bee crawls through that chamber the following season to emerge, the pollen mites will connect all over her body. And you can see in this picture how many pollen mites are on the back of this poor little bee. It's even under the wings. You can't really tell, but this little bee is not going to survive because of all these nasty pollen mites. So if you're hosting your own bees, it's really, really important to harvest and clean every year. The old school way of putting blocks out where you drill holes in it or use bamboo reeds, that's the old way of doing things. We now know with research and by doing this for over 60 years that if you don't harvest and clean, you're actually going to be creating a predator home, not a mason bee home. Predators will eventually take over. As you can see in the pictures in the previous slide, how many predators are inside each cell. Well, just picture that over time, because they don't go away. The Houdini fly will hatch and go out, but the pollen mites will still stay there, and the fungus from the chalkbrood will still stay there as well. So the best type of nesting material to use are like stacking trays that you can pull apart. That's what we use in our blocks, or you can use cardboard tubes that you can unravel and clean. So before I show you the research lab, I wanted to share with you a little bit about what we're finding when we open up our nesting blocks. But I wanted to show you, this is, we opened up a whole bunch of nesting blocks today, and I wanted to show you a little bit about what we're finding inside. Now, if you haven't watched the part 1, part 2, part 3 series of What Are Your Baby Bees Doing, check out our YouTube channel where we open up the blocks and show you exactly the larvae state to the cocoon state. It's pretty cool to see. But as we're examining our bees today, we've noticed that, again, every season this happens, we have so many pollen mites. So these are little pollen mites. And a lot of people love to host their own mason bees. And if you do, it's so important to harvest and clean. Because what happens in the spring when this is a fully grown, well, the bee will emerge from this little cocoon, and he'll crawl, or she or he will crawl through the chamber, through the pollen mites out to get out. And as it crawls through, these little pollen mites will stick to the back, and it will eventually kill the bee and then also spread pollen mites all over the flowers. The other thing that we're noticing are the Houdini fly larvae. And again, these guys will stay here all winter long, and they will emerge with the mason bees in the springtime as well. And they eat the pollen loaf that the mason bee left for its baby. And then the Houdini fly larvae will then eat all of it, and it will kill the mason bee. And you can see here, these were little baby mason bee larvae that have CHALKBROOD and fungus on them that has killed them. So it's so important to harvest and clean all of your mason bee cocoons every fall. I know I mention it over and over again, but it's just so important to reiterate how critical it is to harvest and clean your cocoons, because these predators can decimate our solitary bee populations. So as you can see in the video, there are hundreds of little pollen mites, CHALKBROOD, and Houdini fly that take over those nesting cells and really harm our solitary bees. So we were really excited to be working again with UC Riverside. They contacted us because they are doing a new study on predators, specifically the pollen mites in CHALKBROOD. So we collected samples and put them in vials, and we sent them over to the research lab at UC Riverside. And then we got to talk to PhD students, Jessica and Jacqueline, in the entomology group. And we learned that Jacqueline is going to be handling the pollen mite research. And pollen mites are kleptoparasites. When I asked her what they're going to be doing, she is so passionate about describing how she's going to be researching pollen mites and what she's going to be doing. And I'm going to share with you some of the things that she's discovered. But let's talk a little bit about what she is looking for. Pollen mites are found in orchard bee nests where they feed on pollen provisions and they impact the bee health. Her research is focused on how the mites are being distributed through the nest, how they may be transferred from nest to nest, and she wants to get a better understanding on their biology. So she's going to learn more in depth on how they are using those bees as a host, and she wants to figure out a way of making sure that we can lower the bee mortality in osmia nest. Osmia is the scientific term for mason bees. So there's Osmia lignaria, Osmia cornifrons. There's a lot of different versions and species of Osmia. So that is the study that she is doing on our little mason bees, pollen mites. So you ready to see some slides? So the first slide that she sent me were microscopic pictures of pollen mites. They are so tiny you can't see them with your human eye, but you can see them with your human eye, but when they're all clustered together like you can see inside the nesting blocks and then on top of the bee, they are so tiny, like little mites. They look like crab under the microscope, and they multiply very, very rapidly. All right, I'm going to show you a video of what they look like underneath the microscope. As you can see, they're the small, there's the big ones, there's little eggs in there, and they multiply quite quickly. There's quite a few. You can see how fast they move around. Fast under the microscope. You don't see them move around when you're looking at them in the nesting blocks. But if you happen to take a little patch of pollen mites and you put them on a piece of paper, they will eventually spread out. They move quite quickly. So you can really see how they move underneath that microscope. So a little bit about what she's learned about pollen mites. I asked her how many pollen mites are in each one of those cells, because when we're harvesting and cleaning them, we just see so many pollen mites. And she said there are thousands of eggs laid, and they burst into little mites in each cell. She did a little study where she took 20 mites and she put them in a microcentrifuge tube, and in less than a week there were 100 more pollen mites. She said they are strategists, so they produce a lot of offspring in an unstable environment. She says that the key to their success are they are able to withstand more extreme environments and they don't feed. She did a study with one male osmia where she introduced the pollen mites to a male mason bee, and she put it in the freezer for a few days. The bee died, but the mites still survived. So knowing this information is one way that we can learn how to rid them and rid the predators and make sure that they're not harming any more of our solitary bees. That's one of the things that we learned is when we sterilize our nesting blocks over flame, that kills the pollen mites. It also kills the chalkbrood. But with research teams like Jacqueline, we're able to learn even more about these harmful predators so that we can help our solitary bee population. And then Jessica, she did a study on chalkbrood. And chalkbrood is a fungus that bees will go and collect just in their habitat, in their nature. And then they carry that fungus back into the nesting chamber and the holes that they lay their babies in. There are different species in this group. Some of the chalkbrood or fungus species aren't bad for the bees, meaning that they're non-pathogen. And then the other species are the ones that harm the bees, meaning they're called pathogens. And in Jessica's research in the McFrederick Lab, she was studying the harmful pathogens by comparing them to the non-pathogens. She wanted to find out what the difference was between the two, because some are harmful, some are not. So what's the difference between the two? She also wanted to take a look at how the bee and the pathogen that causes chalkbrood have been co-evolving together. So just like humans, they are made up of DNA that is organized into genes. And by studying the genes that make the pathogens different from the non-pathogens, so the good from the bad, the genes involved in the bee and their co-evolution. So she wants to understand the evolution of the chalkbrood and the fungus. And so she's able to take a look at the DNA to figure out what is harming the bee. So it's pretty fascinating what she has uncovered. And the whole goal of her research is to make sure to aid in the development of treatment for chalkbrood or prevention of chalkbrood, which would be so wonderful to have. If we were able to start figuring this stuff out, we could really help our solitary bees. So we really appreciate the research that both Jessica and Jacqueline are doing to learn more about these harmful predators. All right, so let's take a look of what Jessica found under the microscope for chalkbrood. So as you can see, these were little baby mason bees who have contracted this fungus called chalkbrood. And to me, under a microscope, it looks like they've completely dried out. This fungus started off on them as a teeny tiny larvae state, and as they grew, it just grew with them and it multiplied, and it kills every single larvae that gets this fungus, this chalkbrood. They do not survive, they die. And that fungus will remain in that nesting chamber. It won't go away. The only way to get rid of it is to sterilize it, which is again why we always please ask that you sterilize and clean your nesting blocks. So this is pretty fascinating to see that the chalkbrood under a microscope it looks so damaging. As you can see by the research team and what they've discovered, it's pretty amazing to see them in action and what they're working on. And I just wanted to wrap all of this up. We do partner with a lot of university and research team. It is our mission to maintain healthy mason bees. We want to educate and teach our hosts on how to provide healthy and safe habitats so that our solitary bees can thrive in their environment and in their habitat. that. And if you are interested in hosting your own bees, please make sure you are using the proper nesting material. You're harvesting and cleaning them every fall. If you want to just release solitary bees back into the environment and rent the nesting blocks from us, that's part of our rental program. So you're welcome to go to our website at rentmasonbees.com and check that out. Please follow our YouTube channel and our Facebook page, where we post all sorts of really great safeguarding tips and really great information on our solitary bees. So I'm sure I'll get some more results from Jacqueline and Jessica. They've just been doing this, I think, for a couple months now. So if we get any more research results from them, we will be sure to share them with all of you. And I'm excited because I've been talking to a few other research groups. One group is studying the soil and the clay and which type of clay those mason bees are going to pick. So she has a sample of a whole bunch of different variations of clay. And then I'm speaking with another research team that is working with our host. Well, actually our bees at our host. But they are going to be studying the pollen and what they're eating and the environment in which the mason bees are in and what they are choosing to use for their food. So lots of really great information of learning and research on our little solitary bees, and we will always make sure to share all of this with you because it's important to bring you along this journey with us. Because if you're one of our hosts or you're a solitary bee raiser of your own, it's important to know how we can care for all of our pollinators. So thanks for checking in today, and please stay tuned. We will always have more stuff, and we love to teach. So call us if you have any questions. Thank you so much. Bye.